Hi, I'm Adrienne Nelson, and this is my local expert Module 4 presentation over AlphaFold, which is used to predict protein folding patterns. So first we'll talk about what AlphaFold is and why you should care about it. AlphaFold is an AI system that was developed by researchers working in Google's DeepMind lab. It uses computational biology to predict protein folding and structure based on just the amino acid sequence of a particular protein. For over 50 years, researchers have discussed the protein folding problem, and AlphaFold provides a potential solution. So much of medicine and biotechnology rests solely on these evolutionary nanobots called proteins, from biotech research, engineering microbes, to producing new proteins for bioremediation, to creating new and effective drugs to treat a multitude of ailments. Most clinicians and researchers agree that AlphaFold is a powerful tool that will be at the forefront of novel molecular biology research. The idea is simple, even if the execution is not. AlphaFold requires an input of a genomic sequence and gives you an output of an associated protein structure. In the image to the right, we see the molecular image of the human nuclear pore complex, which was predicted and visualized using AlphaFold by Jan Kaczynski's lab at the European Molecular Biology Laboratory in Germany. This was a large undertaking as the complex contains approximately 1,000 protein subunits. They regulate transport of materials between the nucleus and cytoplasm, and being able to predict their folding patterns can help us understand more about how they are able to ferry materials across the cell membrane. So why are we talking about this in MIP 611? Well, in Module 4, we learned all about protein analysis and expression, and just really proteins in general. We covered protein-protein interactions, which is one of the reasons why we need technology like AlphaFold. This AI can help us predict protein structures, and hopefully in the future be a little bit better about predicting protein-protein interactions, as well as drug interactions. We also discussed immunofluorescence in Module 4 as a way to visualize proteins and how they behave in situ. AlphaFold can assist with identifying protein structure and folding, which, when combined with immunofluorescence, can give us more information about a particular protein of interest. Finally, we've discussed a lot about the various properties of proteins, such as molecular weights and how this affects things like column purification of a lysate. So AlphaFold can give us more information that can make protein purification easier when you have an estimate of the various properties in that protein. Other methods of detecting protein structures are listed here. The first one I'll cover is X-ray crystallography. It is probably the most well-known method of determining protein structure. For me, Photo 50 Run by Rosalind Franklin was my first exposure to this technique, and I only recently became aware of how daunting and in-depth crystallography can be. The most difficult, costly, and time-consuming part of this process is trying to get a good crystal so that you can actually analyze it and bombard it with X-rays. Using AlphaFold, you remove this issue entirely because you don't even have to be in the lab to use it. You just need computational power. Fluorescence spectroscopy is another method used to determine protein structure, but it also requires a lot of skill regarding laboratory techniques and practices. When you're labeling a protein for spectroscopy, you can also have the potential to change the protein entirely, and this can mess up your whole experiment. Finally, we have talked in class about next generation sequencing methods for proteins, and you can use these to discover the composition of samples, as well as see different variations in amino acids, post-translational modifications, and just changes to the overall sequence. I think that with AlphaFold and NGS combined, we'll be able to discover more about various proteins, and this will be a solid tool when you use them both in tandem. This slide contains a figure from the linked manuscript at the bottom of the slide and discusses the current coverage of the human proteome. The Protein Data Bank, or as I will refer to it, the PDB, contains the last 20 plus years of work regarding protein structures. In 1995, there were less than 5,000 protein coordinate files, and as of 2020, we now have over 170,000 files. This is thanks to the contribution of scientists over the years, and now we have a much higher coverage of the human proteome, as evidenced in the figure to the left, specifically A, B, and C. The current coverage of the human proteome is only 48%, as evidenced in figure G. And while this is a pretty significant process over the last 20 years, AlphaFold can help us increase that coverage in the future. This figure highlights how AlphaFold has helped us increase coverage of proteins with high confidence. In figure A, we can see a significant increase from the left to right figure, showing that after AlphaFold was released, we have higher coverage of proteins. Prior to AlphaFold's release, figure B highlights the number of human proteins that had less than 1% coverage. This figure shows us that AlphaFold is most important in predicting structures of proteins that have a high degree of uncertainty regarding their structural status, rather than proteins that we already have a lot of coverage for. AlphaFold is a powerful tool, but like anything, it's not perfect. One of the current limitations is that it is not very good at predicting how the protein will interact with other molecules, such as novel drugs, recombinant protein, and native molecules in the cell. It also can't predict the effects of mutations, so if you want to know what will happen if several amino acids are removed from the sequence, AlphaFold really can't help you right now. AlphaFold also requires a proficient knowledge of not only computational biology, but computer science as well. Currently, it is only available to run on a Linux operating system, and Linux is not super user-friendly like Apple's or Windows operating systems, so you have to have a certain degree of technological literacy and knowledge of programming to conduct this technique. I attempted to install and run AlphaFold on my own PC and found it extremely daunting, and I gave up. While I consider my PC to be fairly powerful, you do need a spare 3 terabytes, that's spare, of space just to store the genetic databases that AlphaFold uses to make its predictions. My PC only has 2 terabytes total, so I was already out of the running for that. Beyond available disk space, AlphaFold requires a lot of processing power in the form of graphics processing units or GPUs, and most recommend the higher end NVIDIA graphics cards and multiples of them. This can get really costly really quick, as even a high-end last generation NVIDIA GPU can run over $1,500. Ask me how I know that. Thankfully, if you can get AlphaFold to run, it does have some built-in controls. That 3 terabyte genetic database contains all of the known protein structures that have been confirmed through experimental results in the past, 30, 40, 50 years. And this helps train the AI model, making it more accurate in predicting protein structure. Finally, AlphaFold's accuracy was assessed using the global distance test, and that measures how a predicted structure is similar to an experimental structure derived in lab. Finally, I will discuss a really not so efficient way at predicting protein folding, and that is high school students. So based on my own experience, AlphaFold can predict protein structures thousands, if not millions, or billions times better than current high school students ranging in ages from 16 to 18. For my genetics class, I gave students a strand of DNA that was derived from the human hemoglobin protein, and I didn't want to overwhelm them, and just because we only have so much time and we're almost at the end of the semester, they were only given 15 codons from the entire sequence, and I think the entire sequence was close to 600 amino acids. And so based on polarity and the charges of individual amino acids, students had to fold their paper proteins into various loops and structures. I think the activity was a success because afterwards students reported feeling confused, overwhelmed, and baffled regarding protein folding, and that's pretty accurate. So thank you for watching my presentation on AlphaFold. Here are the websites and videos and manuscripts that I referenced. I hope in the future that AlphaFold will be more accessible to people that are not proficient in computer language and programming, but it would have been cool to show an actual tutorial of how to use it, but this will have to do for now.